Good afternoon, everyone. Welcome to the Worldwide Neuro Theory Forum. My name is Tim Vogels. I shall be your host today for Annegret Faulkner, who is visiting us from Princeton University. Before I introduce Annegret, I shall quickly remind you there's a chat bar on your right that you should feel free to utilize to shoot the stuff and ask questions that you want me to ask Annegret. And you can also ask more complex questions with the Ask a Question button in the bottom. You should feel free also to follow us on Twitter for more upcoming neurotheory extravaganza. And with that, I should introduce to you Annegret, who did her undergrad at Oberlin College and then went to Columbia University to do her PhD with Mickey Goldberg, where I met Annegret a good 1 billion years ago now. And then Annegret went on to do a postdoc with Dae-Yu Lin at NYU, where she picked up her interest in behavior and aggression, and the work that she did there propelled her into a faculty position at Princeton University, where she is currently an assistant professor and currently being actually locked down, doing...
We're less locked down, we're in phase 2.
No experiments. Well, tell us about your previous work, and thank you for coming.
Thanks, Tim, for having me. So I'm going to try share screen here. Okay, let me know when you see me.
Lovely.
Okay, great. Can you see my cursor?
Yes.
Okay, yeah, thanks, Tim. I really am excited to be part of this really exciting symposium. It's been an amazing series of talks, and I really kind of thank you for sort of bringing us through this pandemic in a kind of elegant and intelligent way. So as Tim said, my lab mostly focuses on social behavior, and I'm going to talk about some work that's kind of spanned my postdoc and into my new lab, where we're really interested in what's happening in the brain when animals are socially dominant and when they're defeated. And of course, we're not just interested in the neural dynamics that underlie these processes. We're really kind of spending a lot of time thinking about how to quantify these behaviors. And before I move on into my talk, I just want to take a moment to highlight the photographer of this amazing photograph. I think the photographs that we use in talks, we don't give enough credit to the photographers, but I really love this photo so much. And I read that the photographer, this guy named Sam Rowley, actually spent a week kind of camped out in the London Tube to get this photo. And it won, I think, what's a pretty prestigious wildlife photography prize this year. And I think this illustrates just perfectly how the behaviors that we study in the lab are incredibly ethological, even and I think the dynamics of this shot, where you look at it and you immediately kind of identify with either the aggressor or the defeated individual. It's just perfect. Okay, so the question that my lab really focuses on is how experience, and in particular kind of social experience, reshapes future behavior, and also the sort of underlying neural dynamics. And the sort of one example that I think is really familiar to aggression researchers is this idea of the winner effect. So the winner effect goes like this, right? If you win a fight, and this has been shown in mice and a bunch of other species, if you win a fight, this makes you much more likely to win successive fights, because somehow this facilitates the expression of these future dominance behaviors. And then animals enter this kind of positive feedback loop where experience really reshapes the behaviors that they perform later. And of course, there's a counterexample to this. It's called the loser effect, where now if animals become defeated, this suppresses the expression of these future dominance behaviors and actually puts them in these negative feedback loops. So these positive and negative feedback loops, you can also think about in terms of that they are a persistent internal state. And sort of one way we think about this internal state is that it's called social rank. So if you enter this positive feedback loop, you might be called dominant. You're in a dominant internal state. If you enter this negative feedback loop, you might be called subordinate. And these persistent internal states actually have really real consequences for individuals. So dominant individuals obviously have more opportunities for access to mates and sometimes to prime resources like territory and food. And these often end up having kind of health protective effects over the lifespan. And animals that enter this negative feedback loop, they have fewer opportunities for mates and food. And it's been well described through a number of lab and ecological paradigms that animals kind of enter this sort of persistent negative affect state that I'm going to talk about. So today, I think for the bulk of my talk, I'm going to talk about some work that's really spanned my postdoc and my new lab, where we're going to kind of hang out in this positive feedback loop and really ask how the brain kind of generates and implements these positive motivation action loops. And then I'm going to show you some new data from the lab, and this is definitely kind of a work in progress, where I'm going to focus on this idea that there's actually a lot of individual variability in this negative feedback loop. So some of these animals seem to show these negative affects while others do not. And we're going to take a little bit of a deep dive into kind of understanding what drives individual variability in these defeat-induced internal states. So that's going to be a little bit rough. I'll kind of let you know when I transition into that. It will probably be obvious. Okay. So the circuits that we focus on in the lab are really not focused circuits that I think neurotheorists spend a lot of time thinking about. It's this network called the social behavior network, the left side of this Venn diagram, most of this yellow and green part. Don't worry, you don't need to memorize any of these acronyms except if I tell you to. So it's this subnetwork of areas that are highly recurrent and very interconnected, mostly in the amygdala, the hypothalamus, and the midbrain. And it's important for a lot of reasons. Not only is known for its role in social behaviors, but it has these very complex interconnections with the mesolimbic reward systems. But not only does it do things that involve driving social behaviors, it's involved in kind of changing metabolic expenditure and endocrine profiles. And these circuits are highly, highly conserved, both at the sort of connection and at the sort of anatomical preservation. And we see that the preservation of the circuits is extremely conserved along the anterior-posterior axis of animals, all the way from telostatic fish, shown here at the bottom of the plot, up to mammals. So these areas are tiny, they're at the bottom of the brain, and we really don't know that much about them in terms of dynamics. So this network that's called the social behavior network, for our purposes today, we can really just kind of think about this as the conspecific aggression network, because all of these areas that have been implicated in a range of social behaviors, including sort of maternal and paternal behavior and mating, they've really been explicitly identified as being involved in conspecific aggression. And this data comes really from two separate lines of evidence. So on the one hand, when animals fight each other, we see a high amount of immediate early gene activation across all of these areas inside the circle. So I'm talking about CFOS here, so it's a really slow time scale, so it doesn't really capture anything about the dynamics. And then the second line of evidence that we've known, I don't know, since the 1950s, is that you can put metal stimulating electrodes kind of anywhere along this anterior-posterior axis and get different aspects of conspecific aggression. So it's been known that these areas are functionally involved in the generation of aggression. And although all this work is really important, I think, and then the piece of data that kind of brought this into the modern age was Dayu Lin's demonstration. She was my postdoc advisor at NYU. But during her postdoc with David Anderson, she was the first person to show, using new tools like optogenetics, that you could stimulate with light this tiny area. It's actually a subnucleus of the VMH. It's called the VMHVL, that I'm going to talk a lot about in this talk, that you could actually get animals to attack not only a conspecific male, which you might expect, but also targets that you might not expect. You can get them to attack a female in estrus. And then I think most infamously, they demonstrated that you could flip on the light and get animals to attack a rubber glove. Okay, so despite, I think, all of this beautiful work, it really doesn't give us an insight into what's happening in this network. Sort of how does this network generate these behaviors? And so now, with the advent of all of these really nice tools where we can record from these tiny nuclei in genetically specialized ways, we can really start to ask the questions that we want to ask as good systems biologists. So we can now start to ask, like, what kind of computations are being performed within each node? How is social information being transformed across these nodes? And how does experience change these processes? And so that's kind of the broad direction of my research, and I'm not going to get to all of those questions today, but that's where we're headed. Okay, so to ask the first question that I posed today, sort of how does the brain generate these positive motivation action feedback loops, we have to kind of back into it, because first we actually have to figure out how we really want to quantify aggressive motivation and action. And this is kind of not a trivial task. I think people who don't think about social behaviors don't actually think about how complex these behaviors are to quantify. And although there is some stereotypy in the way that animals, in particular rodents, where this has been quantified, there's been a lot of work in rats and mice, kind of delineating the structure of these, there's still a lot of variability both within animals. And it's very difficult to think about how we might delineate these phases of aggression, because these behaviors, including things like approach and these sensory investigation behaviors like sniffing, are very fast and overlapping. The identification of these behaviors, which has been typically done by hand, is extremely subjective. And if we want to think about this from a neural dynamics perspective, the sensory information that the animal's experiencing as it kind of moves through these loops is constantly changing. And of course, right, there's a lot of individual variability in the execution of these behaviors. So I've spent in my postdoc and now a lot of time trying to think about how we might approach this quantification problem. And we've approached this from a number of different ways. And so in the first way, I've really taken a page from the B.F. Skinner playbook, and we've really tried to operationalize this idea of social motivation. So can we separate out these motivational processes as separate from the action themselves? And then also in my postdoc, I developed these techniques that lean heavily on small muscle EMGs, so we can record more precisely from the kinds of muscles that animals use when they attack each other. And then recently, we've been really kind of going all in with using some new tracking approaches. And this is just an example from our lab where this is using the LEAP developed by the Murthy and Chaffetz lab. I'll play this for you again. We're actually really happy with the tracking that we're getting, even in animals that are identical. And we use both Deep Lab Cut and LEAP for different things. And I'm going to tell you about, at the very end of the talk, about some ways in which I think we're using this tracking to pull out some exciting and new things. Okay. Any questions, Tim, before I move on into the data? Can everybody hear me?
You're doing great. Thank you very much.
Awesome. Just thought I'd check in. Okay, so as I said, the piece of data that got me interested in this whole question was this experiment that Dayu and David did, where they were able to drive animals into this weird regime where they would attack a rubber glove. And I saw this data like now probably almost a decade ago, and I was like, well, how is this happening? What exactly is happening when these animals get stimulated? And there's kind of two alternatives here. So one alternative is that really this area, so this is this area called the VMHVL, it's this small subnucleus of a subnucleus of the hypothalamus. It's at the very bottom of the brain. This is a coronal slice of a mouse brain. It's about 5,000 cells on each side. It's very, very tiny. So on the one hand, when we stimulate this area, maybe all it's doing is kind of driving action. You flip the switch and the animals attack. Sort of in contrast, another idea is that maybe it's actually just changing the internal state of the animal. We're driving into this motivational state, which then increases the probability of attack. And the first thing that we did was actually just try and record from these areas, because as I said before, there's really very little in vivo data across this entire network about what these neurons are doing when animals are performing these behaviors. So we thought we would gain some insight just by performing recordings in this area. And we quickly learned that the responses are really complex and this wasn't going to be the way to go. So we found that neurons in this area respond not only when the animals attack, which we kind of expected, but also when they investigate the males and when they investigate females. And then then what we did was kind of fit the responses of individual neurons with a flexible GLM with a number of different parameters and try and pull out the kinds of properties that individual neurons might be responding to. And the parameters that we use involve things like the distance between the animals when they're attacking, how fast are they moving, how fast is it, how much time is it into the next attack. And what we quickly found is that neurons in the VMHVL are extremely complex. They really are fit by a combination of variables that say something about the sensory world, say something about the actions that the animals are doing, and also what we think of as these state variables, these things like the latency from and to a next attack. So we decided if we really wanted to understand what this area was doing in terms of motivation, we were going to need to take a different tack. So this is when I turned to the sort of...
Can I very briefly ask, so the what you're activating is a really complex sequence of events, right? You're not just saying like move your arm or something like that.
Right. Well, we don't actually know what it's activating at all.
No. And so I wonder if you see sometimes if you don't properly place the electrode that you see only sort of fragments of the entire behavior, or if you can activate the behavior partially, or if you see some sort of tics.
That's a fantastic question, and that's not true for this area, but might be true for other areas. So yeah, maybe if I can come back to the possibility of that at the end of the talk.
All right.
Okay. But this area is almost too small to do sort of anatomically conserved stimulation in different points. Although there is some evidence along the anterior posterior axis of this really tiny area that the anterior part of the axis drives more defensive-like behaviors and the posterior part of this axis drives more offensive behaviors, but I think this still needs to be teased apart a little bit more.
So there's no homunculus of moves. You can't play.
There's no homunculus of moves, not here, at least.
Okay.
Okay. So in order to think about how we might answer this question about whether it's driving motivation or action, right, we developed this task that I think now has been adopted by a couple other labs, and we called it the self-initiated aggression task. And in this task, animals we put a 2-port nose poke panel in the cage and have animals choose right or left to poke. And if they poke to the right, we call this the social port. And after a brief delay, animals get access to a submissive male to attack. So this is basically your basic operant task, but instead of getting sort of food or water at the end, they get access to aggression. And this task is entirely self-paced, so animals can kind of hang out in the cage and decide when they feel like initiating the next aggressive bout. So they can really tell us when they're in a motivated state. And then in contrast, right, the null port doesn't really do anything. And then so we can quantify this motivational phase, which is kind of sensory independent from this interaction phase. Okay, so this is what this looked like when I first did it. Everything was all by hand. Here's an animal with a bunch of electrodes down the VMHVL, and he pokes to the right port, and he receives access to this male that he can attack. And then over the course of this study, we trained something like 70-some mice, and we found that about 2/3 of them really like to do this task. So they increase their poke rate to the social port relative to the null port within a few days of training. And then actually there's this third of mice that seems to kind of almost anti-prefer this task. So they kind of will poke a few times and then figure out that it's not for them. And so for the entirety...
Do they always poke and attack, or is there also mice that are poking and then just hanging out? In other words, is it only friends and enemies, or I guess, is it only submissive and dominant? Do dominants always attack, or do they just want to hang out?
Yeah, so we did a few controls for this. So the fight in this is fixed. So the only animal they ever see is a really subordinate animal that they will attack. The animals that don't attack, that will poke and then not attack, are actually pretty rare. And if you quantify the number of trials in which attacks occurred, it's something like 96 or 97%. They almost always attack.
So Ferdinand is indeed rare.
What?
Ferdinand is rare.
Yeah, exactly. Ferdinand is rare. Well, no. Well, the ones that don't want to engage seem to be a significant percentage of this population. But in our lab, we changed the task somewhat and we automated the timing a little bit more. And now we find way fewer of these animals that don't want to do the task. So I actually think these ones who don't want to do the task just maybe didn't understand it. So when you make it easier for them, they all want to do it. But for our purposes, but sorry, to answer your question, we also did some controls where if you train them to expect a submissive guy and then you give them a dominant guy who actually fights back, they stop poking very quickly. they don't want to do it. They don't want to do it if their likelihood of winning the fight is not high. Make sense?
Yep.
Okay, cool. Yeah, please break in and ask questions at any point in time. So we used a lot of tricks in this study to figure out what the role of VMHVL was in this task. And first we wanted to test the functionality of the VMHVL, and we took a chemogenetic approach. I'm sure almost everybody's familiar with this. These dreads developed by Brian Ross' lab, where we can inactivate cells using this synthetic ligand. And you can train, so we trained animals to poke for a submissive male, so they're happily poking along. And then we can inject them with CNO, which will inactivate the VMHVL. And then look how many pokes they do. And it turned out that when we inactivated the VMHVL with the CNO, we saw far fewer pokes. And what was interesting to us is you can take these exact same animals that learn to poke for a submissive male, retrain them to poke for water. So now they're performing the exact same action, the poke, right? But they're getting a different reward. And then we inactivate with CNO, we see no effect. So it does seem like inactivating this little area has an effect that's specific to its motivation for aggression and not for a sort of any old reward. Okay, so as I said, I'm really interested in the dynamics of this, the dynamics of the VMHVL during this task, and that was kind of my whole reason for adopting such a paradigm, is that we have moments in this task where the motivation is separate from the action. And so what we did was go back and record from this area, and now we can really focus on what the neurons are doing during this motivation part of the task in the sensory independent way, and in the action part of the task. What we found, I think, is a surprising amount of heterogeneity in the response properties. So here's all the neurons that we recorded, kind of time aligned between the seeking and the social phase, where we expected to see a lot of neurons that have their peak response during the social phase, but we found a small but sort of significant population that actually has their peak responses during the seeking phase, that are peak activated during this motivation phase. And if we use PCA to separate out these two populations, and then we look at the responses of these neurons as a function of how much time it's been since the last initiation, right? That's kind of a measure of motivation. If you're very, very motivated, you kind of turn right back around and request another trial. We found that the ramping of these neurons actually scaled as a function of how motivated they were. And this effect was entirely absent in the neurons that had their peak response in the action phase. Okay, so in my lab also, we lean heavily on this tool called fibrophotometry, where we can record the bulk calcium of nodes of neurons in a genetically identified way. And this is a really robust way to look at changes in population activity as a function of experience. So we want to see, now knowing that there's a subpopulation of neurons that shows these ramping dynamics, were these ramping dynamics there from the very beginning, or do they kind of emerge over the task training? So we can use fibrophotometry to record across the entirety of the training and then look at how this population changes. So here's just an example of what this kind of data looks like. This is the fibrophotometry trace when animals are performing this self-initiated aggression task, and each of these blue ticks represents the time of the poke. But what's cool is that if you now zoom into the single trial responses here, and I've marked the moment that the male enters with a red dot, you can really start to see that these ramping dynamics emerge as the animal's kind of leading up to this interaction phase. And if we look at whether this ramping was present in the sort of initial phases when it was still learning the task, it turns out that it wasn't. And we see the slope of this ramp increase as a function of training day. Okay, so lastly, we want to do the flip side, right? So if suppressing this area can kind of turn down the aggressive motivation, can we do the opposite? Can we stimulate the VMHVL and change motivation in a positive way, kind of pushing the animals into this positive feedback loop? So here, kind of in the Diulin approach, we took an optogenetic approach where we injected the VMHVL with channel rhodopsin and placed stimulating fibers over the VMHVL.
Anna, I have a question.
Yes.
I'll read it to you. I haven't read it yet. Is aggressive motivation generated only in the VMHVL or brain network, including VMHVL, MEA, or BNST? Whereas I'm a little bit at a loss as to...
Is the BNST involved?
Yeah, I suppose that's the question.
Yeah, so the bottom line is we really don't know, and I'm very interested in this question. So the VMH, as I said before, this network is super interconnected and extremely recurrent. So we know that there are other areas in this network that you can stimulate and get attack, but so far we haven't tried any other areas in this task. So we don't actually know about the aggressive motivation, but it's likely that this area is not acting alone. So the VMHVL receives something like 30 inputs and 30 outputs, and it's almost impossible that these dynamics, that this is behaving alone. Make sense?
Yeah.
Okay.
How far away is this from sensory inputs?
From sensory inputs. It's a couple synapses from olfactory and chemosensory inputs.
Okay.
They come mostly in this feed-forward way through the medial amygdala.
Thank you.
Okay, so what we did here was kind of take the Diulin approach, but now instead of stimulating when the animals are close to each other, right, when they're nose to nose, we're only going to stimulate when the animals are alone in their home cage. And we're actually kind of only give them a little bit of stimulation. It's kind of somewhere between 3 and 5 hertz. And then we actually turn off the stimulation once the animals poke. So they're actually not being stimulated when they're together. And what we're actually looking for here is sort of once we turn on the stimulation, how much time does it take them to decide that they want this aggression reward? And then what we can do is kind of alternate between sort of real stimulation and sham stimulation, which is just the power of the laser is off, and look at the latency of those pokes between those different trials. And here's just an example whole session where this animal ended up poking something like 37 times, where you can actually see that in the sham stimulation trial where it's not being stimulated, there's a lot of variance in this measure. Sometimes the latency is very short, he's very motivated, there's gray dots that are very low, and then sometimes he kind of takes a break and he kind of checks out of the task when he's not being stimulated. However, when we interleave these stimulation trials, the latency for him to request the stimulation is very low, is kind of consistently low. And this effect ended up being fairly consistent across animals and across days. Okay, so just to give you a sense of what this looked like when we first did it, I feel like our video quality has really improved since I did these experiments a million years ago. So here's just an example when the animal's stimulated alone in his home cage. I kind of sped that up because it's really boring when the animal's not doing anything. He's even kind of engaged in another kind of behavior right now, but as soon as you turn the stimulation on, he immediately goes over and requests his aggression reward. Okay. And then one last thing that I think was cool that we added on to this task is that I told you we used some of these small muscle EMGs. So what we did is kind of stitch a tiny little EMG into the masseter muscle that's called the superficial masseter muscle of the jaw, where we can record the bites using an EMG while the animals are performing this task. And then we can look using the EMG to see how many of these kind of high intensity big bites the animals are performing during the attacks. And we can compare the numbers of those high intensity bites in the so even though the animal's not being stimulated, we can say how many bites occurred in the interaction trials that were after the sham trials versus after the stim trials. And it turned out that the stim trials contain more of these high intensity bites. So just giving a little bit of juice to the VMHVL not only sort of increases motivation to go over and request aggression much faster, it actually increased the intensity of those future interactions, even though he wasn't actually being stimulated. Okay, so I think this paper, this data really kind of pointed to the idea that there's a much more complex role for the VMHVL. Although it has a lot of neurons that do respond during the interaction phase, this subpopulation of neurons that seems to drive the seeking behavior is there. So we have to kind of take a little bit more of a complex view of the VMHVL. Okay, so getting back to my original question about how animals implement these motivation action loops, what we've done is kind of elucidate this complex role for the VMHVL, right? It's responding to all of these different things, but we still don't really understand how this signal generates a discrete event, like the action, right? There's somewhere in the brain, there has to be kind of an attack actuator. And next, I kind of decided that I would look for that place in the circuit. And as I said before, when Tim asked about the circuit, the VMHVL is hugely interconnected, not only with nodes in the social behavior circuit that I showed you before, but so another paper from David Anderson's lab, they did this heroic effort using 4 or 5 different kinds of tracing methods. And they really looked and took a extremely deep dive into the anatomical circuit and quantifying both the inputs and the outputs to the circuit. And so I reasoned that probably the place that's doing this actuator is somewhere on this list, but it would be like a fool's errand, I think, to kind of march through the circuit one by one to do this. So I took a kind of a cheap shortcut. My cheap shortcut is that I reasoned that an area that's doing sort of actuator function would be connected to the jaw in as few synapses as possible. And the way that I tried to test this is I used these pseudorabies viruses. They're actually developed right here at Princeton by Lynn Enquist's lab. So they're transsynaptic viruses that you can actually inject in the muscle. And then depending on how long you wait after you inject them, usually people wait between 96 and 120 hours, it will travel back a certain number of synapses so that we can kind of retrogradely look at all of the neurons in the brain that project to a particular muscle. And so I did this, when I did this, I targeted the same muscle that I did with my EMG, right? It's this muscle called the superficial masseter. It's really important for jaw closing. In these same animals, because the VMHVL is excitatory, I used a V-glu2-Cre animal, and then I labeled the projection fields of all across the brain to all these different areas using a Cre-dependent synaptophysin. And then what I did was look for places of overlap between these jaw projecting muscles and these synaptic fields. And to make a long story short, the place with the most overlap between this sort of synaptophysin and the label is this area called the lateral PAG. And so if you look at our box here, I probably would have gotten there eventually if I kind of marched down the list, but maybe it would have taken me a longer period of time. So here in green, right, these are these retrogradely labeled neurons from the jaw overlaid with the synaptophysin. Okay, so then I kind of...
Very quickly, a little bit off-piste, but did you check whether stimulation of the VMHVL during absence of social stimuli could induce alternative forms of behavior, like feeding in the presence or absence of food?
They don't, it doesn't make them feed, but it does make them just kind of a little bit crazy if you stimulate them when they're alone in their home cage. It increases locomotion. It's almost as if they're looking for something. No, it doesn't increase feeding. It doesn't increase mating, except that, so this, yeah, so David has another paper where they stimulate at low intensity and at high intensity in this subpopulation of VMHVL neurons, and they show that at low intensity you can actually induce mounting behavior, and at high intensity you induce attack. Although it's a little bit unclear whether this mounting behavior is a dominance behavior, kind of low-level dominance behavier, or it's a sexual behavior. So that's a little bit unknown.
And the biting, does that happen only in the presence of a subordinate male, or does the mouse already go around, you know, just like chest biting?
That's a really good question. I have all this data that I've been wanting to publish. So yes, so one of the other things that stimulating the VMHVL does is kind of the animals go into this, do what I call bruxing. So you see this very stereotyped kind of sweep in the EMG where they're actually grinding their teeth. And I'm very fascinated by this because there's some theories in evolution that suggest that teeth grinding is actually very adaptive, because what it does is make your jaw stronger for the moment that you want to attack later. So yes, it does. It has some jaw effects independently of the sensory effects. That's a great question, but I haven't published that yet. Okay, so now, okay, getting back to this actuator idea. So in collaboration with a postdoc in Dayu's lab, she did some extremely heroic slice physiology where she confirmed that there's this functional excitatory connection between the VMHVL and the lateral PAG. So we used V-glu2-Cre to characterize this excitatory circuit. And not only did she found that only excitatory neurons in the lateral PAG receive inputs from the VMHVL. She even was able to take this experiment a step further, where in these same animals, she would inject a retrogradely traveling PRV virus in the jaw. So now we can label neurons that project to the jaw in the PAG and then look at to see whether they get functional connections from the VMHVL. And she found the same thing. So really only neurons that were excitatory and had jaw projecting activity got, not only, so a subpopulation of these excitatory neurons that are jaw projecting receive functional input from the VMHVL. So we really have this nice sort of clean excitatory circuit that's jaw projecting that seems like it could perform an actuator function. Okay, so then we kind of throw our bag of circuit breaking tricks at this circuit to see if we can figure out what's different about it from the VMHDL. And again, I took a chemogenetic approach, remember, so we can use these viruses to kind of turn down the activity of this circuit. We inject a DREAD GI bilaterally in the lateral PAG, and we can just kind of have the animals interact and see what they're doing. So as I mentioned before, the role of the VMH is really complex. We can kind of turn down the VMHDL activity, and we get animals to, I didn't show all this data, but the animals kind of stop attacking, they stop poking, their sensory investigations kind of messed up, and some studies have actually shown that they don't interact with females as much either. But in contrast, right, in the lateral PAG, the effects are actually much more specific. So in a saline trial, if this is a male animal, he kind of investigates, he attacks, goes back and forth. Sorry, this video is long. Okay, and then in the CNO trial, we actually find that he still wants to investigate, and he almost seems, his motivation seems to be very maintained, but he just doesn't really attack anymore, which was really interesting to us. And we found that this effect was highly specific. We found deficits only in attack and not an investigation of a female, not investigation of a male. And then we also tried some controls, given that we know that this area has these projections to the jaw. So it doesn't seem to affect animals' ability to eat sort of palatable food. Okay, so now it does seem like there's this difference between what the VMHVL and what the PAG is doing. So the idea is maybe what the VMHVL and the PAG are actually encoding different phases of aggression. So I showed you this plot before. So the VMHVL is kind of doing complex things. We know that the VMHVL responds during attack. This is in red, and during this investigation of males and females. How am I doing on time? Okay, and then if we actually look at the population here, each dot here is a single neuron. The population is kind of non-specific, like with respect to investigation or attack. You actually wouldn't be able to tell at the level of the population whether the animal is attacking or investigating. And if we look at how many neurons are significant for these behaviors, we find a lot of overlap at the individual neuron level. So now let's look one synapse downstream at these neurons in the lateral PAG. We find that the situation looks quite different. So we actually lose these sensory investigation responses to male and female, the blue and the green here, and we find that we find only a big robust activation during attack. And if we look at the level of the population, now we find that it's hugely divergent. We find a large number of neurons that actually prefer attack as opposed to investigate male. So now it seems like it has this sort of hierarchical representation of aggression. We have kind of multiple phases of aggression represented in the VMHVL, and then only the action represented in the PAG. Okay, so, and then we took it a step further, knowing that this area projects to the jaw, we incorporated this idea that we could record from this muscle, the superficial masseter, along with our ability to record from the lateral PAG. And here's just to show you, I think this is actually quite a technically difficult experiment. We were able to get clean recordings of both the jaw and spikes while animals are attacking. It's actually quite difficult to record robustly when animals are attacking. We're still working on improving our yield in these experiments now. And then we just kind of used mutual information to ask whether having this information about the jaw had any predictive power about the activity of individual neurons. And it turns out that in males, when males are interacting, we find a significant amount of predictive power from the EMG, but we don't have that ability to predict when during female interactions. Even though animals are using their jaws, right? They lick the sides of the cage, they do all kinds of things. Adding the EMG adds nothing to our predictive power. Okay, so, and then just one last thing with this, because I'm really proud of these experiments.
I don't understand what that means. What does that mean? What do I take away from that?
Just having the EMG, the EMG is more correlated during male than female. You can think of it that way.
But that's it?
That's it, yeah.
Okay.
Yeah. So it's not that it's correlated with movement or anything. It's actually correlated because attacks really only happen in the male case.
Okay, got it.
We can think of it as a proxy contact.
Sorry.
But we don't actually have the behavior, right? We only have the EMG.
Yeah.
Okay, then we can use the fact that we have this EMG to look at the relationship between the spikes and the peaks of this EMG in a little bit of a more temporal, precise way. So we can pull out these neurons that have this kind of peak response during attack, and then also look whether these same neurons have increased responsivity if we just look at the EMG aligned to these particular, we look at the EMG aligned to the spikes in these particular neurons. And then what we found is that there's a small but significant subpopulation that has these significant spike-triggered EMGs aligned to this, aligned to the spikes. So it does seem like in the lateral PAG, there's a tight temporal relationship between the spikes and the activity of the EMG. Okay, so now I've shown you that there's this sort of hierarchical relationship between the VMHVL, where the VMHVL is doing all these kind of complex things, where there's kind of this bucket of neurons that respond to the sensory phase, this bucket of neurons that respond to the action phase, and this kind of separate pool of responses that I haven't really talked that much about, the response during the female. Now, if we go one step downstream, we find that the neurons are kind of highly selective for the action phase of attack. And the question is sort of, how do we get from here to here? And there's kind of two simple ideas of how this could be true. So in one case, right, we just have a straight up labeled line. So in this case, we just have, maybe it's just actually a small pool of neurons in the VMHVL that are the projectors. And this is how we get from complex to simple. And on the other hand, maybe you actually, all the information is traveling down the pipe, but what's happening is that there's some filtering, postsynaptic filtering mechanism that's creating this action selectivity on the postsynaptic side. So I devised a couple of, sort of experiments to get at these two hypotheses. So in testing the labeled line hypothesis, we leverage this idea that we could record in a projection-specific way using fiber photometry. So we could inject a retrogradely traveling GCamp in the PAG that would end up in the VMHVL. So we can record only from the VMHVL neurons that project to the PAG. And at the same time, simultaneously, we can record from the entire population of VMHVL neurons, right? So we're recording from the projectors and the projectors plus the non-projectors. And what we did was just kind of have animals interact with males and females in an alternating way, and then look at the relationship between this simultaneously recorded signal. And what we found is that the signal magnitude, sort of what's being relayed in this kind of projection-specific way, is not different for the male case, but it is different for the female case. So the amount of signal that's being relayed down this pipe for the female is reduced. So there is some filtering happening to make this kind of a male-specific labeled line. And then we looked at the correlation between these two signals, kind of separating out what's happening when they're interacting with males and when they're interacting with females. We found that the correlation is between the two signals, these simultaneously recorded signals, is much higher when they're interacting with males than with females. So what this suggests is it's sending a much more sort of faithful copy of what's happening down the pipe to the VMHVL when he's interacting with males than when he's interacting with females. Okay, so this suggests that there's at least a
two questions here.
Yeah.
Did inhibition of glutamatergic neurons in PAG affect self-initiated aggression, seeking, or only execution of aggressive behavior?
We haven't actually done the nose poke on this, so it's on my short list of things to do. My strong hypothesis is that it won't affect the motivation.
All right, and is ontogenetic activation of the VMHVL PAG circuit sufficient for evoking aggression?
So it's complex. So the short answer is that it works sometimes and not others, and we still haven't exactly figured out why. So we have done terminal stimulation. In some animals it works, and then in some animals it doesn't. And I'm not sure why. So what we published seems like it's complicated. Yeah, the short answer is no, it doesn't work very well. So the reason for this is, I think that because the output structure of the VMH is so complex, it requires, and what's happening for the actuation, so what the PAG seems to be doing is kind of stitching together potentially pools of neurons that drive individual muscles. I kind of maybe think of these as like innate motor modules. But attack is actually a very complex thing that requires arousal and endocrine change. And so you need the VMH field to kind of yoke all of these things together. So it's not just sufficient to drive the motor circuit.
So where could you initiate fragments of this? Only in motor cortex itself, or do you think what are the sequences here where are the memories for these sequences?
Where are the memories for these sequences? That is a great question. Why is there some stereotypy in the way that animals move through these sequences?
Yes.
Well, my hunch for this would be that it has to do with sensory information. So actually, let me go through this next piece of data, and maybe this will give you a clue.
Okay.
So the next kind of idea is we were wondering how is the PAG transforming information from the VMHVL? And again, we took a fiber photometry approach to this, which I remember we're recording sort of bulk activity from the VMHVL and the PAG. And we wanted to know kind of what's the relationship between the PAG and the VMHVL at any moment in time. And so now we just did the same thing where we can have animals interact with males or females. And the idea that there's some sort of slow filtering mechanism was kind of born out of a kind of a trivial observation of that if we look at the cross correlation between these signals, there's this kind of asymmetric bias in the male signal where there seems to be some slow change in the VMHVL signal that's driving the PAG signal. And this is completely absent when animals are interacting with females or during the baseline condition. So what we did is just kind of try to make a simple model of how the PAG is responding to the VMHVL. So we just kind of fit a time-varying general linear model, and then looked at how well the PAG, how well we were able to predict the PAG signal from the VMHVL signal using different amounts of time backwards in the VMHVL signal. And then what we found is that sort of invariant in the bin size that we used, both the fit that we were able to get in the male signal was better than the female signal, and also the best fit models that we used incorporated back more than 5 seconds of time for the VMHVL. So what we think is happening here is that actually the PAG is actually sensitive to slower signals in the VMHVL that may tell you something about the sensory history of the animal. So as animals approach, the amount of sensory information is increasing, and then this may drive the PAG to respond. Okay, so just to quickly answer the question that I posed, whether it's a labeled line or some filtering mechanism, it seems to be both, right? So it's filtering out some of this female information, but also doing some slow filtering before driving these muscles. Okay. So just to sum up really quickly what I showed you, so the VMHVL, right, is doing something kind of complex. We know there's neurons that respond during the sensory phase, the aggression-seeking phase, and the action phase. And then down, you go one synapse farther, we find that the PAG is very selective for the action part of this response. Okay, I'm actually going to skip. Nah, maybe I won't. But so in my lab now, we're kind of taking this to the next level where we're really interested in taking a broader perspective of this circuit. So maybe this gets at one of the questions earlier about which pieces of this circuit are actually involved in the motivation and action phase. And because this circuit is so large and complex, we need a way to scale this behavior upward. So to do this, we really thought that maybe inhibition was the key to stabilizing these aggressive motivation and action states. And we can use fiber photometry to actually inject a retrogradely traveling GCamp into the VMHVL. And then we can label every inhibitory population of the brain that projects to the VMHVL to ask how these inhibitory signals are changing as animals learn this task. And again, I said we needed to scale up this task, and we also developed a more automated version of this task where now animals are being delivered on a tether. And this allows us to do this in a much more high throughput way, and we're trying to screen through many candidate areas. Many candidate areas and kind of understand how signals are being stabilized. Okay. And then we've incorporated tracking, and we can form more complex models about these things. Okay, and so just to cut to the chase, I'm not going to walk you through all of the areas that we've screened so far, but we do think that inhibition is involved, in particular from areas in the POA and local inhibition, and from the medial amygdala. And if we look at how the responses change over time, we see that inhibition seems to relax during the motivational phase as animals become experienced. So the amount of inhibition that these areas that drive motivational circuits kind of becomes relaxed over time. And there may be small changes in the timing of this activity at the stop of attack with time. Okay, and then I'm going to skip over this part.
You've got time.
Hmm?
You have time.
I have time? Okay.
Yeah.
Well, sure. Well, I'm not going to the end. I have some more stuff. Well, okay, I'll just a nod. We're also scaling way up in terms of the circuit that we're recording from. We're scaling up to record from 12 sites in 2 colors during these tasks. And what we're really interested is targeting some of these genetically defined areas that may provide what we think of as kind of privileged channels of communication with these tasks. So one of the sort of privileged channels of communication that we're really interested in is hormone receptors, and in particular, these estrogen alpha receptors that were identified again in David's lab. So every area that I told you about in the social behavior network is sort of rich in these ER alpha receptors. And we reason that these kind of circuits could have these, basically be these privileged channels of communication that can turn on and off with hormone states. And just to kind of get at these kinds of questions, we're recording simultaneously from multiple points of this circuit across a bunch of different tasks. And we found that both in the baseline activity and in the social activity, we find far more coupling from these genetically defined circuits than we find in the surrounding areas that don't have these receptors. So it really does seem like these sort of sub-networks within the social behavior network are able to transform information around, and now we're looking to see how maybe changes in the routing of this behavior underlies experience-dependent change. Okay, cool. And people always ask about what's happening in females. We're kind of doing the first recordings across this network in females. And the kinds of things that we find synchronize this network in females are just like the things that synchronize it in males. So here's a really kind of angry female who's pushing, every time she pushes away this male, we see that the network becomes synchronized with her with kind of little mini attacks. But I like to add that we're thinking about these things.
Well, okay. So the mini attacks are nowhere near as big as the
they are not. They are not.
So if you crank up the volume, you don't get anything like the male, or you can.
You're asking if you can stimulate the female and get. So these animals aren't being stimulated; they're just being recorded.
I see. Okay. Okay.
Yeah. But you can, in certain contexts, so females have been, so you can stimulate the ER alpha subpopulation in the VMHVL in females, and in some contexts, in particular in lactation, you can get them to attack. But it's far, far more difficult to drive attacks in females. However, they seem to have all of the same machinery. So I think in terms of sexual dimorphism, it's more of a function of degree, not kind.
Okay, we have a follow-up question for that, but I was going to delay that to the end of the talk.
Okay, I can take it now if it's on this, because I'm going to switch gears a little bit.
Okay, so hold on, I have to find it. Here we go. Here, does it mean that sex-specific information for the hypothalamic aggression need to be expressed in its downstream targets, but not just the VHMVL itself?
Can you repeat that question? I'm not 100%.
I mean, I didn't read that properly. Does it mean that sex-specific information for the hypothalamic aggression needs to be expressed in its downstream targets, or just the VMHVL itself? Like, where is the sexual dimorphism?
Oh, so far the dimorphisms that have been described are kind of small. So I won't get into this too much, but the as I told you, so there's a subpopulation of neurons in the VMHVL that responds to female-specific cues. So the main difference between male and female VMHVL is that in the female, in the male, these two populations, sort of male preferring and female preferring, are somewhat overlapped, maybe like 20%, somewhere between 20 and 30%. And in the female, they're completely non-overlapping. So what this means is probably that the kinds of motor activities that males need to drive with these circuits have some functional overlap between females and males. And that makes sense, right? Because the kinds of actual behaviors that a female does during sex and during aggression are extremely different, but maybe for males they're actually less different. So that's our current thinking on that. Okay. So in the very last few minutes, I'm going to talk about some new work, very much a work in progress, where I'm going to flip now to the negative feedback loop and really kind of think about the things that drive individual variability and defeat-induced stress. And to do this, right, we're really interested in this idea that not all animals who get defeated show these negative affects. So in modern parlance, we sometimes call these animals susceptible, these animals that don't show these negative internal states as being resilient. And many studies and a lot of papers and a lot of ink has been spilled thinking about whether animals have some sort of predisposition for how they deal with this defeat stress, right? So maybe some sort of change in endocrine profile, or they have some difference in a genetic or epigenetic signature. We're taking a much more simple approach where we're actually, okay, maybe what's actually happening is that the behaviors that they're doing while they're being defeated predict whether they end up being resilient or susceptible to these negative states. And so to test this, so we want to kind of develop new pipelines for understanding the behaviors that animals do while they're being defeated. Okay, so to do this, and this was spearheaded by this amazing superstar grad student, Lindsay Wilmore, and this is all in collaboration with Alana Witten's lab, the great Alana Witten, and we've been using this paradigm that's well known in the field. It's called chronic social defeat stress, where animals get defeated 10 days by a really aggressive Swiss Webster male. So the animal that we're recording from now is actually black, and the ones that are the aggressors are white. And then they have to kind of live separated from this aggressor for a barrier. And every day they get a new aggressor. So they get beat up for 10 minutes every day, and they have this sort of chronic stress. And then we measure whether animals are resilient or susceptible post hoc by kind of looking at this measure of avoidance. So animals that have not been stressed, they kind of tend to prefer being around this aggressor, but animals that have been stressed do not. So what we see is that these are animals that have never been stressed. They kind of don't go through this paradigm in the same way. They really do prefer this aggressor, but there's a lot more variance in this stress population that's been repeatedly defeated. And there's actually quite a bit of bimodality in this distribution of how socially avoidant they are. So remember, this happens on kind of like day 11. And so we can split this into two populations that I'll use to divide for the rest of the slides, right? That we call this resilient population that kind of looks a lot more like the control population that was never stressed. And there's this super susceptible population that really kind of has this chronic negative affect states. Okay. So this is post hoc designation as resilient or susceptible. And so Lindsay did kind of an amazing thing, is that now we're kind of going to use some of these tracking methods. We use deep lab cut for this. And in tandem, we're also recording from 2 different brain areas. And I'll tell you about what we see in this. And we're going to take a deep dive looking at the behavior during defeat and the neural dynamics to see if we can find signatures that predict this resilience or depression-like phenotype. Okay, so we use both kind of supervised and unsupervised approaches to look at this behavior. So for our supervised way, we hand labeled a subset of this data. Remember, this is a ton of data because it's kind of 10 days of recording for a bunch of animals. You need a large population of animals to kind of see these effects. So we track the mice, and then we actually have a high school student and some other people who help us kind of hand label a bunch of different behaviors in this data sets, and then we extract features from this tracked behavior, and then train a random forest classifier on the rest of the data. And so here in black, this is the true behavior for each of these behaviors that got labeled, and here's how well our classifier does. In addition to kind of like shortcutting our ability to identify behaviors in this also provides these discrete events that we can align neural activity on. And then our unsupervised approaches, that is still a bit of work in progress, but we're really excited about. We again take this tracking data, we put it into a feature space. We use something like 12 to 15 features. That include things about like the proximity, again, the velocity, some angular information. And then we take an approach that's become, I think, very popular, pioneered by Gordon Berman and Josh Chaffetz, where we now embed this feature space and then segment using watersheding. Okay, so now what kind of data can we get out of this approach? So now we have this post hoc designation of whether animals are susceptible. And so if we look at our supervised approach, we found that we can look at the amount of time the animals spent being attacked, and there's no difference. So animals that were resilient or susceptible were both attacked the same. But animals that seem to employ this fight back strategy, it was far more likely to be employed by resilient animals. And if we look at how this behavior evolves over time, what's interesting to us is that it's not apparent on day one. It's only as animals kind of figure this out, the resilient animals, that this behavior emerges over the 10 days. So it's not as if animals are predisposed to fight back. Okay, and so what kind of information can we get out of
Sorry, there's some evidence that sort of risk-seeking behavior in humans depends on whether their initial risk-seeking behavior is rewarded or not, right? So people that do something stupid and get away with it do it more than the people that on their face, loosely speaking. Have you looked at whether the success rate of the fight back, is there a way to quantify whether the fighting back is actually a useful strategy the first few times around?
Yeah, well, that's exactly what we're looking for and why we're looking at dopamine circuits. In fact, we're kind of trying to look at which behaviors end up being rewarded. So, okay, so, but let me, I'll talk about the individual variability. Oh, I'm sorry, I'm over so we can look at using our unsupervised strategy, and then we can kind of, what we want to do is cluster the data and see if there are unsupervised behavioral clusters that are more enriched in resilient animals as opposed to susceptible animals. And when we kind of do this analysis, we find that there are. We find resilient enriched clusters, and they're different from susceptible enriched clusters. And just to kind of show you what these behaviors look like, right? If we take a look at a susceptible enriched cluster, and we look into this, we can kind of peek into this black box of this cluster. What we see, sort of maybe to the uninitiated, this is happening too fast, but what's happening to this black animal is that they're specifically being attacked from behind. So it seems like maybe they're not able to fend off these rear attacks as much. And then just to look at an example of a kind of behavior that's in a resilient cluster, this doesn't look like anything's happening, but that's kind of the point, right? So in this, it turns out that this resilient cluster actually corresponds to a state when the animal's far away in the cage, but facing the aggressor. So this is a behavior that we might not have pulled out with our supervised approach. So we kind of think of this as maybe sort of a quiet, vigilant state. So animals both need to employ these active strategies and these more passive, vigilant strategies in order to become resilient. So lastly, I'm just going to kind of quickly go through the sort of preliminary data where we're now doing what Tim just suggested. We're kind of looking into these dopamine circuits, and we've targeted two areas that we know are involved in signaling reward and threat. And one is this projection to the nucleus accumbens, which is involved in signaling reward, and the other is this area that's been sort of newly characterized as responding during social threat. And what we're doing is kind of, we're just taking a first pass by looking to see whether there are individual differences in the dynamics of these areas in these resilient and susceptible animals. And what we found, I think, surprisingly, is that if we just look at the activity when the animals are being attacked, overall, animals have more dopamine when they're resilient than when they're susceptible, which is interesting. But then when you look at the responses themselves, the dynamics, we find there's huge differences in the variance, where susceptible animals seem to show this sort of negative RPE when they're getting attacked, but resilient animals are much more variable. In fact, some animals even seem to like getting attacked. And we think this has something to do with the kind of strategies that they're employing. And the story is a little bit more complex in the tail striatum, but overall we see sort of more and less varied threat response in these resilient animals. So bottom line is that these resilient animals sort of show less of this negative reward structure and more threat response during defeat, which kind of goes with our behaviors that we see. So lastly, I just want to kind of point to where we're going with all of this, is that we're kind of capitalizing on our ability and using deep lab cut to do sort of real-time tracking on these animals, and then perform closed-loop experiments using optogenetics, where we can actually stimulate animals while they're in certain clusters to see if we can teach animals to be resilient, right? So that we'll align stimulation when some sort of set of features from the clusters fits into what we call pro-resilient clusters. And we're going to take kind of two tacks on this, which is one, that we're going to use the fact that we can train animals to do things that they like by stimulating dopamine neurons when animals enter pro-resilient clusters. And then kind of to tie back to what I just told you, we're also going to see if we can change the animal's resilience by actually intervening in the behavior, not just teaching them to be resilient, but actually just like get them to perform the behaviors. Okay, so I don't have any data to show you that I feel confident about today, but just stay tuned that this is coming.
Could you short circuit this and just activate resilience like you activate attacking?
Well, yeah, this is strategy 2. So if we just have them attack, does this end up with resilience?
Yeah, or is there a resilience center, so to speak, right?
A resilience center.
Like
yeah.
If you can activate another brain region that makes them more resilient.
More resilient. Yeah, I mean, if there were a resilience center, I mean, we probably wouldn't have to do these experiments. But I do think these are kind of orthogonal strategies, right? One is like you do the thing, and then you say, okay, you teach them to like this thing that is good for them for resilience, and the other one is like you just do the behavior, but maybe they don't like it. So, yeah. But we don't really know yet whether either of these is going to be enough. Okay, so bottom line, if you take away anything from this talk, is that maybe the circuit is complicated, and that I've kind of parsed apart a little bit of this feed-forward circuit from the hypothalamus to the midbrain, that there's just a hierarchical relationship between information that's coded in the VMHVL and the PAG. And lastly, this work in progress that we're really excited about is that we're kind of starting to piece together why there's so much individual variability in these kind of negative states when animals get defeated. And it seems so far that we have both methods to pull out behaviors that differ and dynamics in the dopamine circuits that differ. And just quickly, I want to thank everybody in my lab, especially Tomo, who's gotten the automated nose poke working, Lindsay, who's been working really hard on this defeat strategy stuff, and my postdoc May, who's kind of building some new tech to do these kind of circuit-wide recordings.
And that's it?
That's it.
Yay.
Thank you. Oh, and my collaborators, especially Alana, who's been instrumental in the second part of this talk. Yeah, thank you for having me.
Well, it's been lovely. I invited Giovanna on screen to ask you a question, and here she is. So I'll let her talk for herself.
Okay.
Hi, can you hear me? Okay.
How do I stop sharing here?
I can unshare you.
Yeah, unshare my screen. Hi.
Hi. Hi. So first of all, thank you for the talk. I really enjoyed it. And I was just wondering, so my question was kind of more about the very beginning of the talk where you were talking about the self-initiated aggression task. Mm-hmm. So it just kind of made me think about the possibility of playful behavior in this task. So would you say that perhaps some of the mice exhibited a sort of ambivalent response because, for example, tickling can be seen as a sort of ambivalent stimulus where you get both positive and negative USVs and also responses in rats in particular. So I was just wondering whether perhaps in this sort of task, there was a possibility that the mice were engaging in a playful aggression, and whether it's even possible to delineate playful aggression, playful behavior from just sort of regular aggression. Yeah.
That's a great question, and to my knowledge, there's not a ton of work done on mouse play behavior. And we know from our EMG recordings that there is a significant amount of sort of high intensity biting. So my guess is that it's not very playful. These animals are also sexually experienced and adults. So that also puts them into the high aggression camp. We haven't yet done these experiments in juveniles where we expect to see play behavior, but that would be a fantastic experiment to do in the future.
Okay, thank you.
Thank you very much. We have another question for you from Jose Maria Martinez de Paz.
Yeah, can you hear me?
Yes.
Great. Thank you for the great talk. My question is about the complexity of the PAG that you have addressed, but not really detailed. And in my understanding, I think that dorsal medial PAGs involved in submissive and defensive behavior. So now you have talked about the lateral one as involved in totally the opposite, and that quite surprised me. I don't know if you know more about the relation between both sections, or maybe I don't know enough about it.
Yeah, so the dorsal medial is not as well characterized as the lateral PAG at this moment. The VMHVL also sends projections to the DM PAG, but we actually don't, there are not any known recordings of the DM PAG to my knowledge in social behavior. So that's on my to-do list, and definitely there is a circuit by which you could imagine that there's some intermediate information transform because the DM is kind of hierarchically organized with the lateral PAG. So basically the VMHVL sends projections to both the lateral PAG and the DM PAG, and then the DM PAG loops down to the lateral PAG. So whether it's the same neuron, whether it's two populations of neurons that perform these projections, or it's the same population or some intermediate transformed information, they're both really cool questions that have not yet been answered.
Okay, thank you.
For the dorsal, but you're not talking about so the dorsal lateral PAG has been well studied by Tiago Branco's lab and a couple of other labs. Yeah. Dorsal lateral PAG has a different hypothalamic projection from a different hypothalamic set of nuclei. It seems to be more involved in predator-based fear. So there are these kinds of parallel circuits that seem to be involved in conspecific aggression that are kind of right next door, but anatomically segregated to the predatory and fear circuits.
Also, there are no connections between them?
Not the DL PAG and the lateral PAG have very few connections. Oh, so it's weird, actually. So if you look at the most of this connectivity stuff has been done in rats. It really needs to be redone in the mouse. But there's actually very few connections between the DL PAG and the L PAG. The DM PAG and the L PAG, highly connected. DL PAG. I'm sorry, I'm getting into the weeds a lot of the circuit here, but
I'm following.
They're all great questions. The PAG is weirdly technically challenging to record from. So one of the things we're trying to do is really scale up our recordings. It's also a huge area. So when people say they record from the PAG, it's many millimeters on the many millimeters along the AP axis. So there's probably huge differences in what's happening along the AP axis that have not yet been explored. For how much real estate the PAG takes up in the brain, we know surprisingly little about it.
Thank you very much.
Thank you.
I think that's it for questions, Annegret.
Okay.
So
That was great.
thank you very much. I'll contact you on the other side. I wanted to quickly say we have been organizing for our speakers. We've tried to emulate the experience of a visit a little bit more than just letting you give your talk and then sending you back into the vacuum by organizing one-on-ones. And so far, we've restricted these one-on-ones to people in the lab, or at least people that are known to me, because we wanted to avoid the equivalent of Zoom bombing in one-on-ones. But for those of you who are still listening, in the future, if you're interested in one-on-ones with any of the speakers, you can contact me ahead of time and we'll organize that. And at some point, we'll be good enough to also send delivery food to the speaker. So maybe sometime in the future, we'll be able to emulate the speaker.
My husband still won't let me have takeout, so taking this COVID thing very seriously.
Maybe a little one. All right. Anyway, in the future, not today. But,
sounds good.
Next time you give a talk on Worldwide Neuro.
Excellent.
We'll order it. With that, thank you very much, Annegret.
Thank you, Tim. Thanks for the invitation. This was really great. Thanks for listening.
See you.
Bye.
See you soon.